Yeah, so that's so hello everyone. So that's exactly what I'm going to do. So I will continue where we stopped yesterday, and actually, so the next thing was actually so we were pretty much finished with the basic processing steps in MaxQuant, and so maybe just to remind you how to obtain the software. So there's the maxquant.org webpage where you can download the MaxQuant software itself. We just upgraded all the versions, so versions of MaxQuant and Perseus were upgraded to 1.6.01 now, and so we will see in a minute why we jumped from 1.5 to 1.6. So there's also the perseusframework.org webpage where you get the Perseus software. You can of course just type them into Google and you will be led to these pages. So most of you probably know that we have these help forums, so both for MaxQuant and for Perseus, and these are Google groups which are heavily visited. So it's a few thousand users that are there. So I should just remark maybe that so we are actually not really taking care of these Google groups anymore because it's just too much and too many questions that me and my colleagues would be too few people to answer. So what's happening instead is that actually users like you are helping other users, and so we're very glad and happy that you are doing this actually. So that's working very well. So usually it's other users that are answering your questions there, and usually it's also done in very good fashion. So then we have this bug reporting system here called UTrack. So there you would report bugs. So but you have to keep in mind this is really different. So this Google group and this bug reporting system are really two different things. You go to the bug reporting system if you have a reproducible thing that goes wrong. So either the software crashes, or you think, or you can show that it's actually wrong, or that a number is wrong, or something like this in the output tables of the software. So then if that's the case, then it's nice to report here. So first of all, how one actually should reproduce this bug, so that me or some developer can easily reproduce it, which is of course a prerequisite for being able to fix it. And then also one thing one should keep in mind is, so this should always be done in the latest available version, because it can happen that the bug that you find in an older version is already fixed in the latest version. So that's actually something very important, just to streamline all of this and not to waste time on wrong things, that this should always be reproduced in the latest available version. So that's very important. So source code is available to some extent. So a lot of like basic utility functions for numerical calculations and things like this are there, but also many of the plugins, so that we will hear about later this week, the plugins for the Perseus software. So many of them are also available in source code from GitHub. And then, as you know, we are filming also right now these sessions, and they are actually now live streamed, but also they will be put as movies onto YouTube for watching them later. And there are already many of these movies from the previous summer schools, which you can just watch there. Right, so that's probably one of the bigger news for this summer school, that so we are supporting now all platforms that are out there. So you are probably aware that so far we were only targeting Windows platforms. So this has something to do with the programming model that we have. So we use C# as a programming language, and so as you know, this is something developed actually by Microsoft, which is by itself not really platform independent. However, there are ways to make it run on Linux and Mac, and so one way of doing is going through the Mono framework, and that is the way that we chose. So Mono is actually an alternative to the .NET framework, which runs on all of the platforms. which sounds nice and easy superficially, but it's actually not as nice and easy, we found, because it's actually you cannot just use any C# code and run it on Mono. So you have to do specific things to it so that that will be possible. And so these things include that you have to use a restricted set of commands within your code, because Mono is always a couple of years behind, and so many things are actually not implemented. Many things that you have on Windows in .NET are not implemented in Mono. So that's what I spent most of the last year on, basically, to get the code clean of these commands so that it would actually run on Mono, and with this actually on these other platforms. So far we're actually only testing on Ubuntu Linux. So Mac is more, it should theoretically work as well, but we haven't really tested yet so far. But if it's not working, it should be little things that should be required to change to also have it running on Mac. And so when we are done, so this is all true for MaxQuant and Perseus. So however, at the moment, what is stably working is not Perseus, but MaxQuant, and from MaxQuant, actually not the graphical user interface, but the command line version. So there is actually a command line version of MaxQuant that was already working for quite some time on Windows as well. So you find it if you go into the bin folder of the MaxQuant installation. There's actually an executable called MaxQuant CMD for command line version, right? And so this just takes the mqpar file, so the XML file that contains all of the parameter values as a command line argument, and then crunches through the project, just like the graphical user interface version that most of you know would do, basically, right? But then this is probably not such a big limitation, because for people who wanted to go to the Linux platform, so they probably anyway wanted to use this from the command line. So either starting it from the shell immediately or starting it from another script or something like this, right? So it's probably anyway what you need, basically, this command line version. And so far, I mean, we didn't do too extensive tests because we only finished it last week, these things. But so far, so the data sets that I ran, they ran through perfectly well. The speed is maybe a little bit slower than on .NET Windows. So that's just because, of course, it was all developed first on Windows and optimized there, and then people reprogrammed it on Linux, and then they just tried as good as they can to achieve the same performance. But it's also performing quite well. Yeah, so what kind of implications has this? So that also means that you can go now to, so if your university has a high-performance computing center or something like this, or you have some of these large-scale computational resources that you have free access to, you could also just run MaxQuant there now. So it's probably, in terms of hardware that you have to buy specific to MaxQuant, it's probably a great relief that you don't have to do this anymore now. Also, so some people found that it's actually quite easy now to install it in cloud environments. So I mean, it's really, I mean, you just put the software there and install it and run it there. So it's really no-brainer, basically, to also run it in cloud environments now. So, but we continue working on this. So the GUI version of MaxQuant on Linux. So there are only a few things missing. Actually, it's mostly not working if you have non-standard displays. So if you have very high resolution or very high density pixel displays, then this graphical user interface looks really bad on Linux. But you see, we are really working on the fine things to finish it. So the GUI version should also be out very soon, I would think. Okay, then, so what is in the pipeline? So that's not for now, but coming later, is that we have a MaxQuant version that does load balancing over several projects. So now it does load balancing for one project, right? So you have one MaxQuant job, you run it, and then MaxQuant does a fairly good job of distributing all of the parts of the workflow over many cores, right? So that's working well, but then, so this version here would be able to do this then also over many projects. So sometimes you have to actually run several MaxQuant jobs that are not connected into one project together, and that would make it much easier to do this, basically. Okay, maybe that question already on this? Or there's one over there.
Thanks. So sorry, I didn't quite get that. Is there going to be a graphical Perseus interface for Linux as well?
Yeah, it's going there. So that's so this is actually working theoretically already. It's just that when we did it and started it, we found that many things are not properly implemented in Mono. So it still requires some extra work to program around some problems that are still there. But so we're doing this, and so this will be there in a couple of weeks. All right, then, yeah, so what's next? Why is there nothing on this? Okay, that's interesting. There was supposed to be something on this slide. So I just wanted to show you how you call it. So if you are now logged in onto Linux computer, then you don't type MaxQuant, but you type Mono, because that's like the program that you actually start, and then you type MaxQuant CMD as the first command and parameter to Mono, so that it knows which program to run. And you might have to specify the full path for this, depending on your environment variables. And then the MQ file, the XML file with the parameters that you actually want to use. And that's all. And it's running. Okay, that's interesting. Okay, but here there is something on this slide. So now we actually continue with the algorithmic stuff that we stopped with in the previous part of this MaxQuant basic session. And so I'm going now through a couple of important features that are, belong to the standard repository of MaxQuant algorithms, and that are needed for, all kinds of steps in making your data good. So one of these is the requantification step. So what is this? So this is where we are now back to the MS1 labeled data. So for example, Silec data with heavy and light pairs on the MS1 level. And there it can happen that, or it for sure happens, that some of the isotope patterns in your data are not paired into heavy light pairs, right? So they stay alone as features where it's not clear if it's a heavy or light peptide per se. And what we do with those is we still want to identify them, and if they have an MSMS spectrum attached to them, then this MSMS spectrum will be submitted to the Andromeda search with the labels as variable modification, because we don't know what it is, so we have to put them in as variable modification for this search. And then assuming that we then identify this peptide here as something, then from the identification we know if it's heavy or light, basically, right? And then in this case, let's say we found that it is a peptide with a heavy lysine from the MSMS identification, then we would actually now make a second attempt to do a ratio quantification for this peptide. So then we would know, so based on this, knowing where the isotope pattern is itself and what the identity is, based on this we know where the partner should be if it's actually there. And then what we do is basically have a closer look at exactly these positions where the partner is supposed to be. And in this case, you see actually that there is something just emerging from the noise, which looks like the partner. And then so these features are also collected, and one does the quantification against these, basically. So the requantification comes with some special considerations that one has to take. And so that is, so first of all, it's prone to underestimate the ratio. And so the reason why it likes to underestimate the ratio is what often happens here is that, I mean, in this cartoon here, you have the situation that, so you have the light form of the peptide identified, so we don't draw now isotope patterns, so just one peak. So it's abundant peak for the light form of the peptide. But the heavy form would actually be below the limit of detection, right? So that would be a case where we don't find a pair in the first steps of MaxQuant. However, then there are two things that can happen. Either there is nothing there even after requantification, or it could also be that, of course, there could also be that something is above the limit of detection, but let's assume now the real peak would be below limit of detection. In this case, it can still be happening that there is just by chance some noise peak there, right? So you're looking at a specific position, and because the data is not completely noise-free, it could also be, even though there's not supposed to be anything, there is actually some signal that you pick up when you look at this particular position. And in that case, you would then actually underestimate the ratio. So underestimating meaning the absolute value of the ratio is smaller then, right? So let's say if the real ratio here, if you were able to detect this one here, would be 20, but then if you detect the noise peak instead of this one that is at the limit of detection, then you would report a ratio of 10 instead of the ratio of 20. And now, or it's actually 1 over 10 or 1 over 20, if the heavy light ratio, yeah, of course. And now you have to make a decision. So in your experiment that you're looking at, what do you want? So do you want to have ratios reported for proteins that show really a very high regulation, like that? You don't really care how high it is, but you just want to know that it is high, and you want to have a ratio reported for these. In this case, you would switch the requantification on, because that's exactly what happens then, right? So then in many cases, and it's actually quite a few cases where you quantify against the noise, because on protein level, you try this as with many peptides within the protein, and it happens quite regularly that at least one or two of them are quantifying against the noise. So that is the one option. So you're happy with MaxQuant showing you large ratios if it's large, but not necessarily correct ones. And if you're not happy with that, so if you say all of my ratios that I have in the table have to be as correct as possible, then you should switch off or not switch on requantification, right? So that's really, one has to think for a few seconds about this, if one wants one or the other. Okay, so then, so there are ways to make the best out of it. So if you now use the requantification to estimate maybe, okay, so we are not really measuring the correct ratios, but quantifying against noise. So how can we still estimate the protein ratios in best possible ways? And so for that we have introduced this advanced ratio estimation for dealing with the situation where you have a mixture of requantified and actually real measured ratios. So we go through some steps basically to see what is happening in the data. And so the first thing we ask then is, do we have actually already sufficiently many non-requantified features? So if we have enough of those, then we can just forget about the requantified features for the protein and just take the median of the non-requantified features. And the threshold that we have built in here is there should be at least 3 peptide sequences, so not just 3 features, but really 3 features coming from different peptide sequences. So if these are found with non-requantified features, then we just forget about the requantified features, whatever they may be, basically, right? So then we have enough data to actually estimate the ratio on real, on non-requantified data. So that's the first question. So if that fails, so there are not enough non-requantified features, then we look at the following. So we look if there is a correlation between the ratios and the intensities, or actually between the log ratios and the log intensities. And why should there be this correlation? So if you are in this situation here, then it actually depends on the height of the. So let's say you have looking at a ratio of 10 in the protein, right? Then it actually depends on the intensity of this peak here, if you can actually measure this ratio 10 or not. Or if you cannot measure it, then it depends on the intensity of this peak. What would actually be the ratio that you get from quantifying against the noise, right? And through this, you actually then get a correlation between the intensity of the feature and the ratio that this feature reports, right? So we make a simple test with Spearman rank correlation. If there is this sort of association between the ratio and the intensity, and then we cut it off at this particular p-value here. And if it's significant according to this, then we do the fit at this data, and then we actually take the ratio value at the highest reported intensity. So this would be the highest reported intensity. However, we don't take the ratio of this point, but we take the ratio that the model would yield at this intensity value, right, to make it a bit more stable and not relying on just one data point. So that's basically kind of as far as we can, yeah. So you don't want to extrapolate really, because you don't really know where to stop with the extrapolation. But that's basically kind of the what we can claim is the biggest the tightest constraint that we have on the ratio, right? The ratio has to be at least that big or higher, basically, right? Because at some point one would actually see that it should plateau. So if we reach enough intensity that we can resolve the dynamic range of the ratio, it should actually not depend on the intensity anymore, right? So that's why that's the best we can do is in this situation is to take the value of the fit at the highest intensity that is involved. Right, so, and I was already mentioning that one should actually see a plateau if you reach basically the intensity that is enough to resolve the dynamic range that you want to measure. It should actually flatten off to exactly the ratio that is actually, that this protein actually has. And so we also make a fit with this hypothesis. And then we compare these two fits and then decide, taking the degrees of freedom into consideration, decide what is the better fit to the data, and then either one or the other is taken. Right? And if this is the best fit, then obviously this plateau value here is taken as the ratio. Yeah. Maybe questions to this? Okay, so, and you control all this by switching on and off advanced ratio estimation. That's the parameter to look for in MaxQuant, and it's actually on by default. Okay, so this was something important for MS1 level labeling. Then so now to something completely different. Match between runs, which is actually important for label-free quantification and MS1 labeling and MS2 level labeling as well. And so the idea here is that you want to basically increase the coverage of identified features that you can use for quantification over many samples. And so if you have identification of a feature in one or few of the samples, but not in others, that just by the presence of the MS1 feature you transfer this identification, and all these MS1 features can then be used for quantification. And that applies both to the label-free quantification, but also for the label-based quantification that would actually help the situation. And so this is done based on the masses and on the retention time of the feature. So the masses now are very precise because, as you know, we went through this process of making a first search, using this for nonlinear recalibration, then making the main search with tight precursor mass windows, and by now we are actually far below sub-ppm on average on the mass error of these features here, which makes the matching, which helps to improve the fidelity of the matching. So false positives are going linear down with shrinking the mass tolerance window that you have to apply now with the matching, right? So that's then, when using similar samples, working very well. One should not use it on two different samples, like for example matching cellular proteomes to plasma proteome or something like this. This will for sure go wrong. So you will just produce too many false positives in this case. But if samples are comparable, this is a well-controlled way of increasing your quantifiable features in your sample. However, so one prerequisite is that you can also compare the retention time. So I already was mentioning the masses are really in good shape at this point of the workflow. That's, however, not necessarily the case for the retention times, which might have, first of all, a big linear offset for whatever reason, but might also have, again, nonlinear problems in recalibration. And for that, we have then the retention time alignment algorithm in MaxQuant as well. And so this is actually something that you don't need to switch on, but it's actually always done automatically. As soon as you say that you want to use this matching between runs, then it also switches on the retention time alignment automatically. And so this is nonlinear retention time alignment. So yeah, the basic idea you see here applied to just 2 LCMS runs. So a bit similar to the first search that we heard about. One does now like a first assignment of precursor pairs or feature pairs between the 2 LCMS runs by using large time windows. You have to use large time windows now because it's not recalibrated yet, so it could be off by 15, 20 minutes. And then one just makes an assignment of features that have then same mass and are in this time window, in this large time window, and forms pairs by this between the 2 LCMS runs. And then for each pair, you make a cross in this plot here. And so the plot is, on the one axis, it's the retention time on one of the 2, it doesn't really matter which one, but one of the 2 LCMS runs. And on the y-axis, you have the retention time difference between the 2 LCMS runs. And then, yeah, you get a curve like, or you get a point cloud like this. And the alignment algorithm does nothing else but fitting a backbone curve to this data here. And so this is how it's done for 2 samples. Then if you have many samples, so there are 2 things to consider. So one is, first of all, MaxQuant does not necessarily align or match everything to everything, but you can actually specify with the fraction parameter for the sample which things are going to be aligned to each other and or matched to each other. The other thing, so this alignment is actually, so it doesn't know anything about the experimental design. However, it measures how similar your samples are to each other, and it really tries to first align the most similar samples to each other. So it's a little bit like hierarchical cluster. It is actually hierarchical clustering of the samples. So it builds up a cluster tree of all of the samples. And then in this tree, from bottom up, basically it first clusters together the samples that are most similar and then goes to the top. So in particular, it does not use like one designated run that you align everything to or something like this, which is a very problematic concept. If you have like a large complex experimental design, it's really not clear which should be that one single run that you align everything to. So that's not how it's done in MaxQuant. Also doesn't build up one master run out of the runs that it's matching everything to, basically. Okay, so that's the retention time alignment. I have the feature that I didn't take the very last version of these slides that I made this morning because, so there's a question over here.
Thanks. So I wanted to ask, because you're doing this retention time alignment, and for this you need to have the retention, the peaks in the LC with reasonable. You need to have this reasonable shape of the peak defined from your LC ionogram from you, and if you do the DDA, you have this top 15 or something method. The thing is, when the spectrometer does the sequencing, it doesn't measure the LC peak intensity. So you have these holes in the LC peaks when you measure your ionogram, when you look at the ionogram. So perhaps you have optimized the number of the sequencing events between the full scans. in order for the work better? Or
so is it a question about ion suppression or?
No, it's a question about the peak characterization in the chromatography.
Yeah.
Because
how to optimize the number of full scans to optimize the
the full scans to the fragmentation events proportion in order to have the peak shape in LC with a reasonable resolution.
Okay. Yeah, I always hope that the experimentalists have done this already when we get the data, basically. But so I think so. The question is probably how many scans do you need to capture a peak, really, right? And then I would think this is probably on the order of, if the average is 10 or something like that, it's probably good, I would think, something like this. It could also be, from MaxQuant point of view, it can also be lower. I think we could still work with an average of 5, 6 or something like this scans per eluting feature. But if you go below this, I think MaxQuant would really get problems of identifying the problem. I mean, the problem won't occur here, but it will occur much earlier. That it's actually very difficult to just identify these features if you don't scan them with enough data points. So we still report features if there are in only 2 consecutive scans. So that's actually the requirement in MaxQuant, and I think that's even a user-defined parameter. You could set it to whatever value you want, but we still report peaks if they are in 2 consecutive scans, and they have to have 2 isotopic peaks. So that is the minimal requirement for reporting a feature. But of course, the average value should be quite a bit higher than that. So I would think that should definitely be 6 or 7 or something like this.
Okay, thanks.
I was wondering, for the retention times, would you expect that shape? Because like in the 60 minutes, like the difference between runs is higher, right? If you go, yeah. So, or to put it differently, can you use that information to learn something about the LC runs? Because you see, like at 60 minutes.
Yeah. Yeah. Probably one can learn some. I mean, I just measure the shape. I mean, and
okay.
I lack the chemical knowledge. I have no chemical training in chemistry or anything. So probably if one has this, one can learn something from this. But
okay. But I mean, this is in MaxQuant. You can extract this.
Yeah. So the MaxQuant does it, and so you can also reproduce these curves. So for example, you can load the evidence table into Perseus, and then you can just do the scatter plot, and then if you look at particular columns like retention time correction and things like this, then you would exactly see these curves. Basically.
Okay.
Yeah. There's one, I think.
So the advanced ratio estimation is best for silac and dimethyl?
Yeah. I think it's generically for all MS1 labels, doesn't matter which one.
Okay, not for LFQ, you don't recommend?
No, for LFQ. So for LFQ, so this is very specifically for MS1 labeling.
Okay.
I mean, the counterpart for LFQ would be exactly this matching between runs, right? That's also a technique to find the missing peptide in a sample, but it works differently. So.
Thank you.
So you can say actually the matching the requantification is more sensitive because you know exactly the position where to look at, right? And then you can take a featureless approach. You just sum up intensity that is there, right? And that works because you know exactly where to look at. That does not work that well for label-free approaches where you have to find the feature in a different run. Right? And not only that you don't know exactly where it is for whatever reasons, because chromatography, it has a different shape or whatever. So there it would be very dangerous just sum up something somewhere without detecting features. So then for the matching between runs, it's always matching between features that are generally detected in this particular run. Right? So the features to be detected, just not identified. But that's really important to make this distinction between finding features in the same run where you know exactly where to look for, or finding features in a separate run. There's one here.
Can you keep hands in the air? So I know.
It's a question again about the advanced ratio estimation. So you showed two models, a plateau one and a linear one.
Yeah.
Does it have anything to do with the protein abundance? So for instance, if a protein is really abundant, super abundant, you take the plateau one model for fitting, and if the protein is low abundant?
I think the abundant proteins have a higher chance of actually having this plateau model realized, right? But that's not the way how the algorithm decides it. The algorithm decides it by just looking at the fit, just does the two fits, and then whatever is better, taking the degrees of freedom into account, is taken. But it's not using information about the abundance. But surely, for abundant proteins, you have a higher chance of having the plateau model. Yeah.
Hi. So if you have two samples having light, both of them, and then you use requantify, and you have one feature that you found only in the heavy form, and you use requantify to find the light one. That's okay. If you now use a match between runs, you only identify the heavy one. Would it look for the heavy and light in the second one, or just the heavy?
No. So the matching between runs for MS1 labeled features is only for the complete features. So only where you have heavy and light together, they are actually matched. So the only heavy and only light, they are not matched between samples. So they're only requantified within the sample.
So in fact, before the searches, you have to a priori set alignment time differences. However, based on this kind of analysis, you clearly see what are the differences, and even during the LC separation, difference in time.
Yeah.
Could this not be used to, for example, then later determine what you set as an alignment time differences for the match between runs?
Yeah, so you could put it to a large value in the beginning where you don't know what the proper value is, right? And then after looking at a plot like this, it's really easy to look at the plot like this because that's just in the evidence table basically. You could then, for example, come to the conclusion here, okay, a time window of 5 would have been already enough, right? And so if you wanted to, you could then repeat it with a time window of 5 to kill a few false positives. So that's, yeah. Well, no, it's not. No, forget about this. So actually, the time window of 5 is just to find where the curve is actually residing. And so there are two time windows that should start from the beginning. So there are two time windows that are important here. So the first time window is like the large scale. So that tells you where is it actually looking for the alignment function, right? So, or what is actually the largest misalignment that happened? So when did it go, the farthest apart? That could be 10 or 15 minutes or something like this, right? And this is the alignment time window. And then there's the matching time window. And the matching time window is actually the width of this thing here. So the matching time window is maybe 1 minute, can also be half a minute, depending on your settings. And the alignment time window is more something like 10 minutes. Right? And so adjusting the alignment time window will not really change much. So then, you could make it smaller so that it still would find the curve, but the curve will be more or less, it will not affect the alignment really much. However, the matching time window, so this has immediate consequences on the false discovery rate, right? Because that actually goes linear down if you shrink the matching time window. So that is really beneficial if you would adjust this to your data. And you would adjust this to your data by looking at what is the width of this data cloud here. Right. Okay, so there don't seem to be further questions for the moment. So let's go to this challenging. So I'm sorry that I really took the wrong version of the slides this morning, I think. So I wanted to say something about isobaric labeling here. So what I already mentioned, we do this for the normal MS2 one, but also if you fragment further to MS3, that you can also analyze in MaxQuant. And so maybe just a few words, how is the quantification done for isobaric labels? And so what we actually don't do is that we go immediately to ratios to the reference and then take the median of ratios, as we would do for SILAC, for example. So it looks like it's usually better to just sum up the intensities of the channels. So, and that's what we do. So with one further thing we do. So first, the intensities are multiplied by the ion injection time for Orbitrap data, because then we get to the actual number of ions. And so that's basically what matters. So the more ions you have for the signal here, the less is the chance that it's actually contaminated with co-fragmented peptides. That's why this is basically the thing that goes into the as weight into the summarization. So what we do is, so on the peptide or the feature level, we just read out what is in the channels, like in the 10 channels for 10 TMT. This is going to be multiplied by the ion time, and this is just summed up on the protein level, 10 channels, and that's what you then would load into Perseus, for example. So we could also implement alternatives like taking ratio to a reference channel or things like this. But so we have tried this like internally in the software, and it really looked good actually. But I think that's something that people always want to try, and at the moment it's just this summed up intensity what is there. But that's also what we would recommend anyway. There's a question over there.
So what's the normalized intensity versus intensity in different channels?
Normal, the corrected intensity, right? I think it's, so you talked about the TMT channel, the corrected intensity. So this is, exactly, so this is correcting for mixing, like you have this mixing parameters, how one particular channel is mixing in the +1, +2, -1, -2 channels. And the corrected intensities is just taking this mixing into account and subtracting it, basically. And so this is usually, so for small ratios, the mixing is actually no big deal because it's a percent effect. But then if you're measuring large ratios, then the mixing is actually dangerous, basically, for TMT.
So Juergen, in a thermal or in Proteome Discover, what they do is they look at the signal to noise in order to get the isobaric tags. So can you comment on that?
Yeah, absolutely. So we benchmark this. So we actually compared to signal to noise measurement. And so if you look at the channel intensities themselves, of course, they are different. If you now put the signal in there or signal noise, they're of course different. But as soon as you compare, if you compare them relative to each other, there is nearly no difference. So it's almost the same what you get out. So I don't think it's important to go to signal noise. But that would be another option that we should offer, so that all of you can see that him or herself that it doesn't make a difference to do that. Okay. So there's something on this side, fortunately. That's also one of the older ones. So this is just to say what do we do for MS1 level labeling again now, to go to the protein level. So as you heard for the isobaric labeling, we just sum up the channel intensities multiplied by the ion injection times. For MS1 level labeling, we take the median of ratios. So why does one do one here and the other there? Because the very different nature of the data. So because here, with the MS1 level labeling, there is no interference problem at all, basically. So if there was any, because you had like two co-eluting features of the same mass, it had been killed already by the things that we do within the feature to estimate the Silac ratio, for example. So here, actually, as soon as you have a ratio, no matter if it's coming from a low abundant or high abundant feature, it's a good ratio, basically, right? And that's why here it makes sense to just take the median over all of these, because that's the best you can do then, because then you can again have another level where you now kill like outliers, right? And so that's yet another thing that that contributes to the very high accuracy of the MS1 label workflow in MaxQuant. So we take the median, and then as an example, if you have, as soon as you have 3 points or more, and one of them is outlier, the median wouldn't care about. So the median would actually get rid of this outlier. And just as a reminder, that's not what we do in the isobaric labeling, because there, if you have a low abundant feature, it's actually much more likely to be contaminated by co-fragmentation than a high abundant feature. So that's why one doesn't want to put them all onto the same level into the summarization. But actually, the way it comes already as an intensity is already the perfect way to put this information into this game. Okay, so that was the quantification and how it propagates to the protein level. So one very important thing also is getting back to identification and how we actually go from the identification scores of the search engine to identify proteins in the end. And so what we like to use there as a tool is this posterior error probability. So this is first of all because it is a very nice statistical handle on the certainty of your identification. So going from an arbitrary scale of the search engine score, where one maybe has some guideline numbers to tell you what is a good score and things like this, to something that is more statistically qualified. But also, so the other even bigger advantage is that this is an integration tool. So it can actually integrate a lot of the other knowledge apart from the score of the PSM into the identification process. And so the whole thing is target decoy based. So meaning that we use like the true database, sequence database, and automatically MaxQuant generates then an equally sized database of wrong protein sequences by reversing them, and then basically one knows if one identifies one of those that this is a false positives, and that helps then to identify or to estimate the number of false positives in the forward database as well. And so the posterior error probability is just this conditional probability given the score. What is the likelihood that this was a wrong identification? And so I was already mentioning it has so this PEP has the capacity to integrate information, and you do this by making this conditional probability not just depending on the score, but make it dependent on all kinds of other properties of the peptide or of the PSM, basically. And so that could be, for example, the length of the peptide, or the number of modifications in the peptide, or the number of miscleavages, or the charge. So because all of them give you a different a priori probability that this is a good identification. As one can, for example, immediately understand from the number of modifications. So if you have a normal proteome sample and you have, let's say you have two PSMs to the same spectrum, one of them saying this has no modifications, the other saying this has 27 modifications. And even though they might have the same Andromeda score, it's still much more likely that the one saying this has no modification is the correct answer, right? Just because it's very unlikely that on a small peptide stretch you do actually have that many modifications, or if they were there that you would be able to measure them. And so that's why this is an important a priori information that should go into this conditional probability. Same for miscleavages. Also, if you have a normal trypsin digestion and then for a certain PSM you have, let's say, the suggestion that this is completely cleaved, and then you have another with the same score saying this has 27 miscleavages, then it's again by the same logic clear which is the more likely one. Right? So that's why all these parameters go in here in this conditional probability. And so the other way it actually helps to integrate data is that, so this is really makes scores comparable for non-comparable data. So for example, if you compare data with different fragmentation techniques, so then actually the Andromeda score is not comparable. Like an Andromeda score of 100 for HCD is not comparable to Andromeda score of 100 for ETD. So it's not clear that these are equally well identified, because you have completely different statistical background for the calculation of the scores, because you're offering actually different number of theoretical fragments in each of these searches. However, the posterior error probability, they are actually comparable between these parts of the data set, right? So that's then another thing that it does. So it harmonizes the data. You can have a very heterogeneous data in one MaxQuant project, and still all of these can be compared to each other after going through the PEP calculation. And also they can now contribute in equal ways to the protein identification, because they are comparable, right? Okay, so that's posterior error probability. So once we have calculated these, and so this is something you do on the peptide spectrum match level, on the PSM level, we can now easily calculate or enforce the false discovery rate, or calculate the Q value, so this is all equivalent, on the PSM level, right? Because we then just take this posterior error probability as the thing that we sort the table of PSMs by, and then if you want to have 1% FDR there, you just cut the table off when you have 1% reverse hits, basically. So now you can do this on several other levels. You can do this on the peptide level. So, and this is different from the PSM level, because so on the peptide level, that means you would first merge all of the PSMs that identify the same peptide sequence in your data set, right? So that's one level up, basically. And so let's say you enforce a false discovery rate of 1% here, and you do nothing else, and then you look what you have on the peptide level. So this will not be 1% here as well. So this actually will usually be a higher value. So you could have 2% FDR, for example, on the peptide level, when you were enforcing 1% one on the PSM level. And so this is true. So if you, for the other levels where you can enforce false discovery rates, so there are at least two more that I know of. So one is on the protein group level. So many peptides go together into a protein group, and then you can have another level of false discovery rate control there. And again, so if you just enforce something on the PSM level, let's say 1%, if you measure it then on the protein group level, it will be a completely different number. It has nothing to do with each other. The only thing you can say, this is probably going to be much bigger than this one here. And then you could also do it, some people also do it on the gene level. So that just means that protein groups that match to the same gene, you put together, and then you do the estimation here. So what is done in MaxQuant is, so the PSM level, so that's where we did all of these posterior error probability calculations. So that's a natural place where we do false discovery rate control. But then in addition, and that's actually very important that one does this in addition, we also do it on the protein group level, because otherwise, in particular, if you have very large data sets, you get an accumulation of false positives on the protein level. So if you just keep measuring, and you only control FDR on the PSM level, then you are first very happy because you get more and more proteins just by measuring maybe even the same sample very, very often. But soon you will get very depressed, because then if you would then look at what is actually the false discovery rate on the protein level, you will see that it's maybe 100% basically, right? So, so one has to really take care of doing protein FDR. Right, so this just summarizes, I already said that basically. So we have two levels of FDR control. So on the PSM level, so the default here is 1% that we're using. And then followed by that is following that is the protein grouping. So out of the peptides that we identify here, we make a peptide to protein matching, and then proteins that we cannot distinguish based on the peptide features from the sample are grouped together into protein groups. As simple as that. And once we have the protein grouping done, then one needs a quantity by which you basically sort the protein group list. And so this is just the product of, or it's the sum of the log posterior error probabilities from the peptide level So this we then use to define protein group level FDR. And actually, so since some time already, we also report a Q value in the protein groups table, which means you could also first not enforce the protein FDR, leave it open, but then you have a Q value reported in the table, and they could later use this Q value for filtering. So meaning that if you filter the Q value for being smaller than 0.01, that would actually correspond to the protein group set with 1% FDR. So then one aspect is this so-called razor protein FDR, which is switched on by default, which is just saying that one should really use each peptide feature only once. So that is only identifying one of the, so I'm talking now about peptides that are not unique, that are shared between protein groups. Then one should be very careful with using them then actually for identifying all the protein groups that it appears in, because very likely, or maybe it was actually only one of them, right? One just doesn't know. So the evidence is only there that at least one of these protein groups where it occurs in is actually identified. And that's why one should, under normal circumstances, unless one has a good reason not to, one should use each peptide only once. Otherwise, you would, for example, identify like 200 olfactory receptors just because they have one peptide in common, which one should probably not do. So here you just see that, so now we have this Q value option in, so it's just an example over a couple of data sets, that we have this Q value in the protein groups table. And so here we have a couple of data sets where we didn't enforce the 1% FDR, but rather want to plot dependence on the Q value. And then you see here basically that the protein group FDR or the protein level FDR has a huge impact on your coverage. So for example, in this data set here, so 1% FDR would be very low here. You would actually cut the curves where they are just start to climb up. And you see that your coverage actually can be any value between the true one and 100,000 proteins or something like this, depending on what protein level FDR you choose. So it's basically, if you don't apply protein level FDR, it's completely meaningless to talk about coverage at all, basically. Yeah, here just to show that the razor protein FDR that we're talking about in the previous slide is also having a big effect on the number of proteins that you identify. So in this case, it's actually changing the number of identified proteins by 3,000 or something like this. And you are really on the safe side if you use this razor protein FDR. It's also the standard setting in MaxQuant, meaning again that each peptide is only, each non-unique peptide is used only as evidence for one of the protein groups. One has to click somewhere. Yeah, just on the side. Okay, so we're actually at the end. So we already went through internet presence and so on. So that you know already, where you can find further information. Then another thing to announce is we already have found a nice place for the next summer school. It's actually hard to beat this place, I think. I like this very much, but maybe this is actually going to beat it. So next summer school will be in Barcelona. However, not in the cathedral, but at the CRG, Center for Genome Regulation. And if you have been there, then you know it's actually better than the cathedral, because this is just on the beach in the center of Barcelona, so it could not be any better place, basically. And even if the workshop is a complete disaster, I think just because it's there, it's actually going to be good. So yeah, that's it. So thank you for your attention.
there must be for sure some more questions. If not, I can start with one. Will you publish a paper about the TMT in MaxQuant and the comparison to all the different ways in which you can do TMT?
Yeah, that's actually Pili's paper over there, and we're hoping to finish that soon, yes.
Okay, good.
There's one.
I was just wondering how the decoy database is formed. Is it reverse sequences or scrambled sequences or both? Other?
Yeah, default is the reverse. You can also generate, so there's an option to generate scrambled sequences, but reverse is kind of preferable because if you reverse the sequence, they're more similar to real sequences than completely scrambled sequences, because you want the decoy sequences to be as similar as possible to the real sequences, however not exactly the same, right? So, and that's what you achieve much better with reversing than with scrambling.
Why doesn't MaxQuant incorporate some post-translation modifications that are already available in MaxQuant, like sulfonic acid or sulfonic acid, like simple stuff?
Why they're not pre-configured in the why you cannot select them from the menu?
I think it's not available. For example, I was looking for ADP rivalization.
Yeah, so I mean, first of all, you can just define them yourselves, right? I mean, you can just.
Yeah,
we will learn that in the sessions that how you configure new
very
modifications. But if there are important ones missing, you can just drop us a line and we just add them there. Yeah.
Okay, thank you.
There's one in front here.
Sorry.
I'm just wondering how, from a technical point of view, do you combine different kinds of labelings together? What I mean is, for example, if you have, let's say, Silex sample that you also then want to multiplex by TMT, how would MaxQuant manage this?
Because not at all. So that's
exactly.
So that we cannot do, no.
So could you then just define one as, let's say, variable modification and then the other one as labeling? Would this?
That should work to some extent, yes. So basically, the Silex would be, you have to treat them as variable modifications, and then it would quantify all of them with TMT. And you then would probably have to do some post-processing of the output matrices to get really your desired output. But in principle, that would work.
Okay, what would be the, what should one really pay attention to then, to do it correctly and not...
What should one what?
Pay attention to do it correctly when it comes to this post-processing that usually MaxQuant just does.
Yeah, too many things, I guess. Yeah, I mean, you have to, so you want to get to some protein level intensity, your 2 10-plex TMTs or something like this, right? And so these you would probably, yeah, so you could, for example, go to the modified or modification-specific peptide table, and then you have to sort them basically by, so having the same sequence, but heavy and light. And so these are now beneath each other, and they have to be next to each other, basically. So that, I mean, that's essentially all you need to do.
Okay, thank you.
Okay, thanks again, Juergen.